We're going to take a look at polyclonal and monoclonal antibodies and then some agglutination assays. So polyclonal antibodies are antibodies that are made by different B lymphocytes, and they're going to recognize epitopes or binding sites on the same antigen. So let's say that we've got an E. coli bacteria here, and we have 3 different B lymphocytes that recognize that bacteria, and they're going to be binding to different epitopes on the same antigen. So the antibodies that are made, we've got 3 different types in this example. They all will bind to the E. coli bacteria, but they're going to bind to different epitopes or different sites on that same antigen. Here's a picture of one way to make polyclonal antibodies. We're going to inject an antigen. So maybe in this case, maybe we want to make antibodies against strep bacteria. We're going to inject that into a lab animal, a rabbit in this case. And the rabbit's B lymphocyte that recognize the strep bacteria, we will activate them to start dividing. They're going to form the effector cells, the plasma cells, which will make all the antibodies. And then they're also going to form some memory B cells for the next time as well. The reason that these are called polyclonal antibodies is that notice that we have 2 different B lymphocytes in this case that both recognize the strep bacteria. So they both recognize the same antigen, but they're going to be binding to different epitopes or binding sites on the same antigen. And remember that a clone is just a population of cells that are all the same. So we've got 2 different clones here, or polyclonal, and we're going to be producing 2 different types of antibodies. They both bind to the strep bacteria, but they're going to bind to different epitopes or different binding sites on the strep bacteria. And then after a few days, once the rabbit has made the antibodies, we collect blood from the rabbit, and then we can purify those polyclonal antibodies that bind to the strep bacteria. Polyclonal antibodies are used in a lot of different places. They're used in research and also in some diagnostic tests to diagnose certain diseases. And another place they're used is they make up RhoGAM. So remember we talked about giving antibodies to the Rh negative mom to make sure that her immune system does not see her baby's Rh positive red blood cells. So the RhoGAM is actually made of human IgG antibodies that will bind to the Rh factor, and that'll prevent the hemolytic disease of the newborn. So I would start this is important to know about the polyclonal antibodies. Their advantage is that they're inexpensive. So they're going to be much cheaper than the monoclonal antibodies we'll talk about next. The disadvantage, though, is cross-reactivity. So let's take a look at what happens in cross-reactivity. Here's a picture showing cross-reactivity with polyclonal antibodies. So these polyclonal antibodies both bind to the Salmonella bacteria. But the problem with the polyclonal antibodies is because they're not all exactly the same and they don't always bind to the exact same epitope on the same antigen, it is possible for some of these polyclonal antibodies, for example this blue one, to also bind to something else. So in this case, this polyclonal antibody not only binds to Salmonella, but it also binds to E. coli bacteria. So if you had a diagnostic test that you're trying to determine if your patient has Salmonella or not, and if they have E. coli instead, it would show up as a false positive test result. And the test would show up positive, but they don't really have Salmonella, they have E. coli instead. And that's due to this cross-reactivity of these polyclonal antibodies. So they could bind to more than one thing, and you might get a false positive test result. Monoclonal antibodies, which are abbreviated MABs, they're made by a single B lymphocyte, and they're only going to bind to one specific epitope on an antigen. So because they're all made by the same B lymphocyte, all of these monoclonal antibodies are exactly the same. So you can see these monoclonal antibodies over here, they're only going to bind to this triangular epitope on this particular antigen. So the advantage of using the monoclonal antibodies is that there's no cross-reactivity, so you're not going to get these false positive test results. The disadvantage, though, is they're much more expensive to make than the polyclonal antibodies. So monoclonal antibodies are used in a lot of different places in diagnostic tests, research, and they're also being used to treat some cancers, autoimmune diseases, and transplant rejections. To make monoclonal antibodies, they inject the lab animal, here a mouse, with the antigen you want to make the antibody against. And then you'll harvest the plasma cells from the mouse. But remember that these plasma cells don't live very long, only for about a week. But you want to have these cells living a long time so you can keep making more of this particular antibody. So what they do is they fuse this plasma cell that's making the antibody that you want with a plasma cell that's cancerous. And so this hybrid cell that's made is called a hybridoma. And then the suffix oma means cancer or tumor. Remember, cancerous cells live forever, they just keep dividing, and then this cell will keep producing the monoclonal antibody that we want. There's a really nice animation here that explains how these monoclonal antibodies are made, so I'd recommend watching that at home. There's different types of monoclonal antibodies that are available to treat people with. You don't need to memorize any of these, but when you get into your healthcare classes, you will probably see some of these. So we've got some chimeric monoclonal antibodies. So these have a mouse variable region, the tips of the Y's on the antibody, and they have a human constant region. And you can recognize those drugs because they end in Zymab. And then the humanized antibodies, they're mostly human except just for the mouse antigen binding sites, and so their name is going to end in Zumab. And then the fully human antibodies, these are produced by putting the human antibody gene into the mouse, so they make just human antibodies, and their name ends in UMAB. And notice that with these names, the MAB just stands for monoclonal antibody. So the reason that we've done this, the more human the monoclonal antibody is, the less the immune system is going to react to that and cause problems. So these 100% human monoclonal antibodies are the best ones to use because the immune system is not going to react to it. Whereas if you used a monoclonal antibody made by a mouse that's fully mouse antibody, your immune system will react to that. So you have a lot more problems with allergic reactions. So the more human the antibody is, the less reactions you're going to get. So you don't need to know any of that, but just when you get into your healthcare classes, you'll see that. Here's just some examples of using monoclonal antibodies to treat cancer. So you can actually make monoclonal antibodies that will bind to specific antigens that are just found on the cancer cells and not on other cells. So if you give the patient these monoclonal antibodies, the antibodies will bind to just the cancer cells and that will mark them for destruction. And so natural killer cells will come over and destroy those cells. The other thing that they've done is that you can also take these monoclonal antibodies for the cancer cells and actually attach radioactive material to them. So you can actually deliver radiation directly to just the cancer cells and not other cells because these monoclonal antibodies will not bind to other cells. And these two videos here are really nice because they show both of these cancer treatments using monoclonal antibodies. And this is just a nice chart that compares the monoclonal antibodies with the polyclonal antibodies. So make sure you know the differences between them and the advantage and disadvantage of each one. We're just going to briefly touch on the agglutination tests. Agglutinate just means to clump. And then we talked about blood typing before. So we can actually do blood typing for the A, B, and O blood types, and then also for the Rh factor, whether you're Rh positive or Rh negative. And the reason we want to test for that is we want to make sure we don't cause a blood transfusion reaction if we give the wrong blood type. And if you remember the type of hypersensitivity reaction a blood transfusion reaction is, remember that's an example of a type 2 hypersensitivity reaction. And remember, type 2 hypersensitivity reactions are cytotoxic, and it's where the cells get coated with the IgG and IgM antibodies, and that sets off the complement system, and then those donated red blood cells are killed, and that causes a blood transfusion reaction. So I do want you to know how blood typing works, and we talked about this in a previous lecture with these blood typing cards. So this circle we've got antibodies against the A antigen, and then this one we've got antibodies against the B antigen, and then on the lower left we have antibodies that are against the D antigen or the Rh factor, and then this is just our control, and you just look for where the blood clumps, and that means that you do have that particular antigen that's in that circle. So we'll put this on the homework and we'll see what blood type you think is on this card. And then just remember with the blood transfusion reactions, if you have type A blood, you naturally make antibodies against the B antigen. And then if you have type B blood, you naturally make antibodies against the A antigen. If you have type AB blood, you have both the A and B antigen on the surface of the red blood cells, so you're not going to make antibody against either one. And if you have type O blood, then you don't have any antigens on the red blood cell surface, and you will make antibodies against both the A and the B antigens. And then this is just a nice summary on the bottom of which blood types you can be transfused with. And notice that type O is the universal donor. So anyone could have type O blood because there's no antigens on it to react to. And if you have type AB blood, you're the universal recipient. So that means you can get any type of blood because you don't make any antibodies against any of the antigens.